I'll be speedrunning genetic inheritance. Yeah, okay, so first, past paper questions. Coat color in cats is determined by a sex-linked gene with two alleles, black and orange. When black cats are mated with orange cats, the female offspring are always tortoiseshells. Their coats show black and orange patches, but of various sizes, while the male offspring have the same coat color as their mother. So, okay, for A, using the symbol XB for black, XO for orange, draw genetic diagrams to account for both these crosses. So I've actually already write down the parental black female. Okay, as we know, female chromosome is XX and male chromosome is XY. And because it's black, yeah, okay, black female and orange male. Orange male, male only have one X chromosome, so it must be XOY. And the filial, or I write it down as F1, is basically the first filial, or like the first generations, is tortoise female with XBXO and black male, which is XBY. Why is this? Because to produce a daughter, we need to have both X chromosomes from the father and the mother. So the father will will inherit its X chromosome, which contains the orange alleles, and the mom only have this black X chromosomes, and so it will just give this genotype to its daughter, which will be tortoise female, and the black male will of course get its X chromosomes from its mom and Y chromosomes from its dad, and so it is XBY. For orange females, it is XOXO, and the black female is XBY. And its daughter will have one X chromosomes from its mom and one X chromosomes from its dad. So it is XBXO, tortoiseshell female, and the male will have one X chromosomes from its mom and the Y chromosomes from its dad. So it's going to be XOY. And B, list the genotype and their phenotypes of the offspring that may result from mating a tortoiseshell's female with a black male. So I've actually you need to draw the diagram. No, you need to explain, but I will just draw the diagram to have better visualization. So this is the mom. Tortoiseshell's cat basically just have like this patches of different colors, and its dad is black, XBY and XBXO. So the filial, there are two cases, female and male. The male one will inherit one of its mother X chromosomes, which may be XB or XO, and of course it will inherit its dad's Y chromosomes. So this is why the male have two possibilities, which is a black cat or an orange cat. Meanwhile, the female have also have a two possibilities, which is the XBXO tortoiseshell cat. So it received the XB chromosome from its mom, and it receives the XO chromosomes from its mom and XB chromosomes from its dad. And there are also another possibility, which is a black cat, which is receive XB chromosomes from its dad and XB chromosomes from its mom. Okay, so suggest an explanation for the tortoiseshell coat in terms of the activity of X chromosome. So as we know, female mammals have two X chromosomes. Meanwhile, male have one. Females would have a double dose of X-linked genes. To balance this, one X chromosome is silenced in every cell of the female embryo during early development. So it actually quite random. It's called random silencing. The choice of which X chromosomes to inactivate, the one from the mother or the one from the father. It's entirely random in each individual cells. It's called the Barr body if you want to search it out. So one of it will just silence into a tight condensed chromosomes. So if it's condensed, it cannot express the genes. So one would normally be uncondensed, and the Barr body will be condensed and thus not expressed. The genes in this Barr body is not expressed. So that's why there are patches of cells, like creates clones or patches of cells with different color across the cat's skin. Next, this is in my metaphase 1. Why? Because it still has the homologous chromosomes, which means it's of course in the first meiosis. This we need to write if it's first or second. It's first because it still has both the homologous chromosomes, and it looks like it is aligned in a plate, so it's a metaphase. And describe what has occurred between the two homologous chromosomes. As we can see, there is a chiasma where crossover occur, and homologous chromosome pairing, crossover, and this is crossover is where exchange of genetic material between non-sister chromatids occur, and this can lead to variations because when this chiasma breaks, it will breakage of linkage group creates new combination of allele when sister chromatids are separated. After this is separated, it will create new combinations. And two other sources of variations can be from random orientation at metaphase 1 and 2. Subsequently leads to independent assortment. What is independent assortment? So there are a few arrangements that a cell can have. Like for example, the blue one just say it's from its dad, and the red one just say it's from its mom. And there's like this two-way when the cell divides, either they have like the dad and dad, or like the mom and mom, and this one like have dad mom and dad mom. Wait, I hope you understand. So basically there's just like this combinations when the homologous chromosome pairs, like the left one and the right one can be like from its mom, dad, mom, dad, dad, mom, and so on. And we have, and just say that we have n numbers of chromosome of pairs, chromosome pairs. So the total combination will be 2 powered by n. In humans, we have 23 pairs, or like 46 chromosomes, and it result in like this 8 million of possible combinations. Let's continue. As I said, there's 2 powered by n possible combinations when n is number of chromosome pairs, and also there's random fusion of haploid gametes, so like from the mother and the dad. gametes and join together and just creates a new variety. Okay, next, the number of squash. Okay, basically A for white, capital A for white and small a for yellow, B for disc and B for spear. Okay, this is the parental. We just need to make a diagram. Actually, yeah, okay. The parental is heterozygote for both, and this is the gamete produced. And this is a Punnett square. This is called a Punnett square, where we're just going. So we have like these gametes, right? And yeah, it just pair ups and to make this 16 possibilities of the filials. Anyway, we need to write down the genotypes and phenotypes. This is just for visualizations. And the questions it is asked, what is the proportions of offspring for both yellow genes for both wait sorry what proportions will have yellow and spear-shaped fruits, which is both like recessive homozygote, and it's this one. It's 1/16. Okay, let's go. I'm too lazy. Sickle cell anemia is a blood disease. Okay, you read it yourself. Okay, homozygote dominant for sickle cell allele is like a disadvantage because you could die from malaria. Meanwhile, homozygote recessive for sickle cell allele is like you have this advantage because you will die from sickle cell anemia. But the heterozygote carrier, heterozygote and carrier is basically the same thing, have this one advantage that the malaria cannot acknowledge the cell, so it is more likely to survive and reproduce and pass on sickle cell allele. Okay, next. This is a blood group. There is A, B, O, and basically the dominancy of A and B is the same, like same level or of dominancy, but it's more dominant than O. So that's why when you have EA, EB, you will have a blood group of AB. If you have EA, EO, which mean like this is more dominant than O, so you will have A. To have, and also it is the same for IBIO, it will result in B. Meanwhile, if you want to have blood group of O, you need to have both the IO gene, so you have O. And this is codominance basically. This AB is codominance, is where both alleles are expressed and influence the phenotype. So more than two phenotypes possibilities, like you have this AB, A and B. And phenotype of heterozygote different from either homozygote. Okay. Next, in humans, a gene that codes for protein called is located on the dominant. Okay, so son would receive the Y chromosomes from father, which doesn't carry hemophilia allele. So like the hemophilia allele is in the X chromosome. So example, this is the dad, this is the dad, and the son will have X from its mother and Y from its father. So this son won't have the alleles, the hemophilia alleles. But if they have another daughter, which of course X from the mother, X chromosomes from the mom, and X chromosome from dad, which contain these hemophilia alleles. And if this girl marries someone and yeah, and has a boy, a boy, Y from its dad, and half and 50% possibility that it will inherit half of this hemophilia genes, half not. So father will pass hemophilia alleles to daughters, which will be a carrier. Carrier means it's heterozygotes, and may pass, because it may or may not pass allele to her son. But yeah. Next, a gene for father color. So black is CB and white is CW. And there's another gene that is that make this phenotype Barrett feathers. And yeah, so actually in this case it's unique. It's the male is XX and the female is XY. But actually in real life it's actually the ZW system. So ZZ is male, ZW is female, but in this context we're just going to use XX for male and female XY. This is exceptions. It doesn't work like this in all animals, but yeah, gitu. Terus nah, male, black, non-barred feather. So unless how a farmer could use a okay, you can understand it yourself, please. I'm tired. Next, explain how a farmer could use a breeding program to find out genotype of male chicken with blue barred feather. So we know that blue is a heterozygote. And anyway, this phenomenon is called intermediate. So normally when we have like this is, for example, this gene is red and this gene is white. Normally we have like this is going to be red, right? Like the heterozygote will be red, but not in the case of intermediate. This will produce pink. It's in the same as this case, black and white creates blue. Yeah, it's intermediate. So we know that it's heterozygote, so we don't need to find the genotype of the C, but we need to know if it's like XA or like this, if it's homozygote or heterozygote. So we need to test cross. What is test cross? It is crossing with the recessive allele. And if all offspring barred, it must be it must be homozygote dominant. If some offspring are non-barred, I don't know how to read this actually. Yeah, it must be a heterozygote. I hope you understand. Because, okay. So if ini test cross, this is the test cross. It will produce like XA. Ya, gitu deh, capek. Udah, ya, capek. Ini belum deh. Okay, easy number. How to distinguish between phenotype and genotype? Phenotype is the feature or characteristic result from interaction of genotype and environment on organism. Can be altered by the environment. Meanwhile, genotype cannot be altered by environment and it is genetic characteristic inherited. So it is inherited from your mom, from your dad. And there are two selections, artificial and natural. Of course, from its name, artificial selection is carried out by human. Like they just choose which benefits them, and the gene pool will favor the one that they try to select. Yeah, natural selection is carried out by environment, and gene is length of DNA or sequence of base or locus on chromosomes, and it is coding for a characteristics. Allele is an alternative form of a gene. You guys need to memorize this. Yeah. Done. I'm tired. Bye guys. Thank you.